Epigenetics is the study of how genes are controlled without changing the DNA sequence itself. Epigenetics refers to heritable changes in gene expression that don't involve alterations to the DNA sequence itself. Think of it as modifications on top of genetics that influence how genes are read and used by the cell. Like bookmarks or highlights in a book, epigenetic marks determine which parts of the genetic code are read. Epigenetic changes determine which genes are active or inactive in different cells, leading to diverse cell functions and characteristics. That's how cells with the same DNA can become nerve cells, blood cells, or skin cells. There are three main mechanisms driving epigenetic changes: DNA methylation, histone modification, and non-coding RNAs. Let's look at each in detail. DNA methylation involves adding a methyl group to a DNA base, typically cytosine. The methyl group attaches to the cytosine base. When methylation occurs in promoter regions, it often silences gene expression, effectively turning the gene off. This is one way cells can turn genes on and off without changing the DNA sequence itself. Histone modifications affect how DNA is packaged around histone proteins to form chromatin. These modifications, such as methylation, acetylation, phosphorylation, and ubiquitination, can have different effects on gene expression. Some modifications open up chromatin, promoting gene expression, while others condense it, repressing gene expression. This process is crucial for controlling which genes are accessible for transcription in different cell types and under different conditions. Non-coding RNAs, like microRNAs, regulate gene expression by interacting with DNA, RNA, or proteins. For example, microRNAs can bind to messenger RNA and prevent translation into proteins. Epigenetic changes have several key features that distinguish them from genetic mutations. Unlike genetic mutations, epigenetic changes are often reversible, offering potential therapeutic targets. They are generally stable through cell divisions and can sometimes persist across generations. Environmental factors like diet, smoking, stress, and exposure to toxins can induce epigenetic changes, highlighting the dynamic interplay between our genes and our environment. Environmental factors can trigger changes to our epigenome. Diet, stress, toxin exposure, and smoking are just a few examples. These factors can lead to modifications like DNA methylation, altering how our genes function without changing the underlying DNA sequence. Epigenetics adds another layer of complexity to our understanding of genetics and disease development, illustrating how our genes are not our destiny. Recent technological advancements have revolutionized epigenetics research, allowing scientists to investigate multiple epigenetic marks on a genome-wide scale. These technologies now enable researchers to study epigenetic modifications at the single cell and even single molecule level. RNA methylation, particularly N6-methyladenosine or M6A, has emerged as a significant player in epigenetic regulation. This modification is especially important in the cellular stress response, allowing for rapid gene expression changes. Protein glycosylation, which is the addition of sugar groups to proteins, plays a central role in DNA methylation. This process is key to understanding the mechanism behind this fundamental epigenetic modification. Studies have revealed that epigenetic changes caused by exposure to trauma can be passed on to children born after the event. This transgenerational inheritance means that children of trauma survivors may inherit epigenetic modifications that affect their gene expression. These inherited epigenetic changes have been linked to increased vulnerability to mental health conditions such as PTSD, depression, and anxiety. A classic example of epigenetic inheritance is the agouti mouse, where epigenetic modifications to the agouti gene affect coat color and health. Yellow mice have an unmethylated agouti gene. This leads to yellow fur and health problems like obesity and diabetes. Brown mice have a methylated agouti gene, resulting in brown fur and normal metabolism. Interestingly, the mother's diet during pregnancy influences the epigenetic state of the agouti gene in offspring. The Dutch hunger winter of 1944 to 1945 provides a powerful example of how environmental conditions can cause epigenetic changes in humans. Studies found that individuals who were exposed to the famine while in utero experienced lasting epigenetic changes. These individuals showed altered DNA methylation patterns and increased risk for health issues like high cholesterol, obesity, diabetes, and even schizophrenia. Research has shown that epigenetic changes caused by trauma in Holocaust survivors can be passed on to their children. These changes affect genes linked to stress responses and mental health, such as the FKBP5 gene that regulates cortisol levels. Children of Holocaust survivors show higher vulnerability to stress-related disorders like PTSD and depression, even though they did not directly experience the trauma. In cancer, epigenetic alterations are heavily implicated in tumor development and progression. Cancer cells typically show global hypomethylation, meaning a decrease in overall DNA methylation across the genome. At the same time, cancer cells often show hypermethylation of tumor suppressor genes, silencing these protective genes and promoting cancer growth. Smoking has been shown to cause specific epigenetic changes that can persist even after quitting. One of the most well-documented changes is decreased DNA methylation at certain regions of the AHRR gene in smokers. Interestingly, when people quit smoking, their DNA methylation levels gradually increase at these sites, though it may take years to return to levels similar to those who have never smoked. As our understanding of epigenetics advances, we're increasingly aware of how environmental factors can influence our genetic expression across generations. These discoveries are revolutionizing how we understand inheritance, disease risk, and the long-lasting effects of environmental exposures on human health.